Hello, everybody. In this lecture, we're going to talk about the structure of DNA. Specifically, we'll discuss what DNA is made of, how the bases pair together, and why DNA strands are described as anti-parallel. Understanding DNA structure is important because structure determines how DNA stores and copies genetic information. And so, by the end of this module, you should be able to describe the biochemical structure of deoxyribonucleic acid or deoxyribonucleotides, identify the base pairs used in the synthesis of DNA, and explain why the double helix of DNA is described as antiparallel. All right, now understanding this information is going to help you a lot when you get into the actual DNA replication process because it all plays an important role. And so here we see a individual deoxyribonucleotide. You can see the phosphate group, which is that P surrounded by the O's. Then you can see the deoxyribose sugar, which is that little that little ring that has the five corners on it, and then the O in the middle and the nitrogenous base, which is up top there and just says base. These nucleotides are the building blocks of DNA. The phosphate and sugar form the outer backbone of DNA, while the bases contain the actual genetic information. So you can see here that the carbon is numbered. We number our sugars our deoxyribose or ribose sugars, like 1 to 5. The terminology is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. You can see the little prime notation next to the numbers. This numbering becomes important later when we discuss DNA directionality. Now, you can see in the 2 prime carbon, it's missing an oxygen. It just has the hydrogen. In RNA, on the other hand, you have an oxygen there. And that's why this is deoxyribose. Ribose has an oxygen in both positions. Deoxyribose only has a oxygen in the 3 prime position. That missing oxygen helps make DNA a little bit more chemically stable. Having the extra oxygen makes RNA a little bit less stable. Now here we can see the four nitrogenous bases found in DNA, adenine or A, thymine or T, cytosine or C, and guanine or G. Notice that adenine and guanine have larger double ring structures, while cytosine and thymine have smaller single ring structures. The larger bases with the double rings are called purines, and the smaller bases with the single rings are called pyrimidines. Now notice also that every pairing is a single ring paired with a double ring, and you never have two double rings or two single rings. This is important for keeping the width of the DNA molecule approximately the same. If you had double ring, single ring, or pairs, then you would have these bulges out at the double rings where it'd be fatter, and these jumps in with the single rings where it'd be skinnier. Now here we see the complementary base pairing, like if we look at the same image here, adenine always pairs with thymine, cytosine always pairs with guanine. These pairs are held together by hydrogen bonds. Notice that the AT pair has 2 hydrogen bonds and the GC pair has 3 hydrogen bonds. Therefore, GC pairings are a little bit stronger. They have that one extra bond. This specificity in pairing is extremely important for allowing DNA to be copied accurately during DNA replication. Now here we see the nucleotides connect together to form a strand of DNA. The phosphate group of one nucleotide binds to the sugar of the next nucleotide, creating the sugar phosphate backbone. Notice that the strand has directionality with a 5' end and a 3' end. The direction is based on the numbering of carbons in the sugar, right? So if we look from top to bottom, we see that 5' carbon, which is going to be connected ultimately to a phosphate. We go down to the 3', which is going to go to another phosphate, to the 5' down to the 3, to the phosphate, to the 5, to the 3. And if we just do that ad nauseum until we get to the end of our DNA molecule. So DNA has a 5 to 3 directionality. All right, so the double helix, this is because we have these two individual strands of DNA that are twisted together and they are bound together with their hydrogen bonds between their nitrogenous bases. And when we form this double helix, the phosphate backbone stays on the outside of the molecule while the nitrogenous bases point inward and pair together. This helps protect the genetic information stored in those bases with that, strong outer backbone, which is covalently bonded, kind of pushing out away from or, towards the environment. Here we can see the anti-parallel structure of DNA. The two strands run in opposite directions. On the left-hand side, we see that top phosphate and then the bottom OH group. So it's like 5 to 3, 5 to 3. Now on the right-hand side, it is the opposite. It goes 3, 5, 3, 5. Now, whenever you pair up your DNA, you're always going to have one running 5 to 3. The other one is anti-parallel, meaning it runs in the opposite direction. All right. Now, hopefully this will help you kind of understand DNA replication a little bit later when we start talking about how that works. Because replication happens in opposite directions. It always goes in the same direction, 5 to 3. But since they're anti-parallel, it's going, basically anti-directional when they're moving. They're always moving 5 to 3, but won't confuse you anymore. We'll look at that when we get to DNA replication. So this is actually necessary for proper DNA structure and function, though. The opposite strand orientation allows the bases to line up correctly for their hydrogen bonds. If you see, if it were turned around, they would not line up properly. We would have, for example, the oxygens connecting together, which would actually not form hydrogen bonds. It would form covalent oxygen bonds, and that would make it a lot harder to bind. So if you look down at that guanosine or guanine and cytosine, we see that if you had actually had flipped those around, you'd have oxygen-oxygen binding, which would not be good for hydrogen bonding. All right. So to summarize, DNA is made up of nucleotides that contain a phosphate group, a deoxyribose sugar, and a nitrogenous base. Those bases pair specifically with A pairing to T and C pairing with G. And two DNA strands twist together into an anti-parallel double helix that stores and transmits genetic information. With that, thank you so much for attending this lecture, and see you at the next one.